I'm talking about how the blood-brain barrier can be involved in limiting drug distribution to the tumor, and in particular to the growing edge or the invasive part of a brain tumor, likely GBM. The blood-brain barrier is comprised of a variety of cell types in your brain capillaries, and it keeps drugs from transporting from the blood into the brain parenchyma or from the blood into the tumor site in some areas. The tumor is highly heterogeneous in terms of its blood-brain barrier. Some areas can get drug, some areas do not. It's those areas that do not get drug that we're really concerned about, obviously, because then obviously even an effective drug will not kill the tumor. One way of getting around it, I think, is going to be developing new drugs that have better blood-brain barrier permeability from the onset. So for instance, we saw this morning in our keynote speaker had a beautiful drug, talked about all the mechanistic issues and how they really figured out the biochemistry involved. However, as a caveat, he said, and of course, it doesn't cross the blood-brain barrier. You can't have everything in life. Well, we'd like to have most things in life so we can preserve life, and in case of getting an active drug across the blood-brain barrier, that's important. So I would say from a chemical synthesis standpoint, we need to do a better job of taking that particular attribute into consideration. Other ways of doing it is, we know mechanisms that limit drug delivery across the BBB or blood-brain barrier, and those include transporters like efflux transport proteins. We can make sure that our drugs either are not substrates for these efflux transporters, or we do something to overcome it, like an inhibitor of the transporter itself. These would be strategies. Other strategies include nanoparticle or liposomal delivery mechanisms or shuttles across an intact blood-brain barrier. These are fraught with a variety of issues. The foibles include things like payload, how much drug you can actually get across using these methods. In my estimation, where greatest impact will be felt will be in the design of the molecule itself. Well, we're working on both GBM, glioblastoma, and brain mets from things like lung and melanoma cells. And we're working on trying to do essentially the pharmacokinetic, pharmacodynamic relationships in a variety of PDX or patient-derived xenografts that recapitulate the tumor in maybe a more realistic way than many other models, and trying to do screens of various compounds, and even within a series of compounds, look at those structures that would have the greatest possibility of activity because they have the greatest penetration across the BBB. This includes both delivery and potency. So if you have something that's potent enough, you don't need to get much in, but if it's not that potent, you need to get more in. So it's a simple equation, but hard to solve. We're undergoing a phase 0 trial right now at Mayo Clinic in Rochester, Minnesota, where guided by various imaging techniques, particular MRI, on initial resection of GBM, we're looking at different regions of the MRI and seeing how much drug penetrates in these different regions, and that will give us some idea about choice of drug. We have two issues at hand. One is delivery, and the other is the genetics of the tumor itself. So we need to improve delivery towards the right target, and that should make a difference. In this phase 0 trial, we're actually measuring two compounds that are given to everybody who gets a craniotomy: an antibiotic, which should not get across an intact BBB very well, and an anti-epileptic, which does. So these are given as prophylactic therapy when you have a resection or any craniotomy. And so basically we have a good pair of, in essence, biomarker compounds to determine what's going on with the BBB in that region, which is guided by MRI. So I think that will give us a lot of information for future trials. One key message is that delivery makes a difference. And it's often important to recognize that if we don't know what to do about something, then we just kind of say, well, that's life. Well, it's important, and so we have to take it into consideration. We do all the mechanistic studies and all this other stuff, but something so simple, which may be boring, as the delivery of the compound to the cell of interest needs to be taken into consideration. We need to change our paradigm in how we do not only our clinical trials, but our preclinical trials. And that paradigm should include, right up front, a consideration of the delivery of the compound to the cell of interest. And so that continues to be my key point, and I will continue to make that key point until we start doing that.